transfer system occurs within the mitochondrion, but particularly it occurs on stalked particles. The stalked particles that are found on the cristae. Cristae, these are extensions of the inner mitochondrial membrane, just like you can see. We see the cristae. The cristae are extensions of the inner mitochondrial membrane. Now on the cristae, that is where we find the stalked particles that we are referring to as the oxysomes. They are called the oxysomes. That is where the electron transport system takes place. In brief, what is this electron transport system? We have basically, or the electron transport system is a system that involves the transfer of electrons from one carrier to another, from one electron carrier to another, and in the process leading to the liberation, leading to the formation of adenosine triphosphate. Remember when we said that each NADH2 liberates 3 ATP molecules and each FADH2 liberates 2 ATP molecules. So we are going to see this, how it occurs within the electron transport system. Let us first get familiar with these parts where it is going to take place. The stalked particles of the cristae, we can see the matrix of the mitochondrion where the Krebs cycle took place. All right, we now move to the electron transport system. We saw from glycolysis and from the Krebs cycle that we have reduced NADH2. Reduced NAD then loses its hydrogen atoms. Remember, it is NADH2. So NADH2 loses the hydrogen atoms. When it loses the hydrogen atoms, it then forms NAD. In other words, NADH2 is oxidized to NAD. The 2 hydrogen atoms lost from NADH2 then pass to FAD, just like we are seeing, reducing FAD to FADH2. I repeat, I rewind. NADH2, nicotinamide adenine dinucleotide, reduced NADH2 from glycolysis and from the Krebs cycle, loses its hydrogen atoms, passing them on to FAD, regenerating the NAD to transport more hydrogen atoms. FAD is then reduced to FADH2, that in turn loses its hydrogen atoms. But in the process of hydrogen atoms being passed from NADH2 to FAD leads to liberation of the first ATP molecule. Leads to liberation of the first ATP molecule. So the hydrogen atoms reduce FAD to FADH2. FADH2 then loses the hydrogen atoms. This time, the hydrogen atoms from FADH2 are accepted by coenzyme Q. Coenzyme Q, forming coenzyme Q, which is reduced, which we are writing as CoQH2, meaning reduced coenzyme Q. At this point, we are seeing that NADH2 loses the hydrogen atoms, which are then taken up by FAD, leading to liberation of the first ATP molecule. The FAD is then reduced to FADH2, which in turn loses the hydrogen atoms, This time they are accepted by coenzyme Q, meaning coenzyme Q becomes reduced, forming CoQH2. CoQH2. Reduced coenzyme Q. Now, reduced coenzyme Q, or the CoQH2, then loses the hydrogen atoms. At this point, when it loses them, it regenerates coenzyme Q, CoQ, and the 2 hydrogen atoms, which then splits into protons and electrons. I repeat there. Coenzyme Q reduced, when it releases its hydrogen atoms, they then split into hydrogen ions and electrons. So 2 hydrogen ions and 2 electrons. Now the electrons then move along a series of electron carriers known as cytochromes. They are commonly 3. We have cytochrome B, cytochrome C, and cytochrome A. So they move along a series of electron carriers, that is cytochrome B to cytochrome C to cytochrome A. However, when cytochrome B takes up the electrons, when it takes up the electrons, it now becomes, when cytochrome B takes up the electrons, we now form because we know addition of electrons is reduction. Therefore, we form reduced cytochrome B. Later, cytochrome B becomes oxidized. In other words, it loses its electrons. When it loses the electrons to cytochrome C, oxidation, a second ATP molecule is formed. Meaning the ATP molecule is formed as a result of oxidation of cytochrome B, passing the electrons to cytochrome C. The electrons then move from cytochrome C again to cytochrome A, liberating another ATP molecule, which is the 3rd ATP molecule. In other words, from CoQ, electrons move along a series of electron carriers, namely the cytochromes, which in the process become reduced and then become oxidized. They gain the electrons and then lose the electrons. At the end, the electrons from cytochrome A, then recombine with the protons. You remember the other protons which were formed as a result of splitting of hydrogen atoms into electrons and protons. The electrons moved along a series of electron carriers, the cytochromes. Now from the last cytochrome A, the electrons recombine with the protons. When they recombine with the protons, we now form hydrogen atoms. We form hydrogen atoms that now combine with oxygen. Remember, this occurs in presence of oxygen. That now combine with oxygen, leading to formation of water. Meaning from the electron transport system, if we are starting with the NADH2, we are having 3 ATP molecules liberated. But if it is FAD, starting from FAD, then there are only 2 ATP molecules liberated, giving off another product known as water. This means that the process of ATP formation in the electron transport system involves oxidation of cytochromes. It involves the oxidation of cytochromes, leading to formation of ATP, hence a process called oxidative phosphorylation. Oxidative phosphorylation because it involves oxidation of cytochromes leading to formation of ATP. But the whole process of oxidative phosphorylation occurs in presence of oxygen. So briefly, we can say and conclude that oxidative phosphorylation is the formation of ATP in presence of oxygen. Presence of ATP in presence of oxygen. In summary, electron transport system. NADH2 loses the hydrogen atoms passed to FAD, in the process liberating the first ATP molecule. FAD then combines with these hydrogen atoms forming FADH2, which in turn loses the hydrogen atoms, and they are taken up by coenzyme Q. Coenzyme Q then becomes reduced to coenzyme QH2, which is CoQH2. Coenzyme QH2 then loses the hydrogen atoms that split into hydrogen ions called protons and electrons. Electrons pass along a series of electron carriers known as cytochromes. That is cytochrome B to cytochrome C to cytochrome A. In the process, more ATP molecules are liberated as a result of oxidation of the cytochromes. Remember, oxidation is the loss of electrons. From cytochrome A, which is the last electron carrier, the electrons recombine with the protons, forming or generating the hydrogen atoms, 2 hydrogen atoms, which in turn combine with oxygen, liberating or forming water. We have said that this process of ETS involves oxidation of cytochromes, leading to formation of ATP in presence of oxygen, a process known as oxidative phosphorylation. Oxidative phosphorylation, which briefly is the formation of ATP in presence of oxygen. Flashback. In your nutrition, photosynthesis, you looked at formation of ATP in presence of light. Formation of ATP in presence of light, which you called photophosphorylation. Photophosphorylation. Here we are seeing formation of ATP in presence of oxygen. Oxidative phosphorylation. Therefore, in respiration, we are talking about oxidative phosphorylation. In nutrition, you talk of photophosphorylation. So that is the electron transport system. Electron transport system, whereby each NADH2 liberates the 3 ATP molecules, and each FADH2 liberates the 2 ATP molecules.